Hello, my name is Nyla King, and the topic I've been assigned to talk about is co-immunoprecipitation and immunoblotting. The presentation will include a background and introduction on the principles of the techniques, some uses of the techniques, a question I have related to the topic, and concluding with a reference slide. Okay, so with the background slash introduction on the principle of the techniques, co-immunoprecipitation is also known as co-IP, and immunoblotting is also known as Western blotting. Co-immunoprecipitation basically purifies a protein or a molecule that's of interest with other molecules bound to it to form a multimolecular complex, and immunoblotting is done by stabilizing a mixture of proteins and separating them according to size. Co-immunoprecipitation is performed in vivo while immunoblotting is done in vitro. So that's a key difference between the two. Okay, on to the next slide. Okay, so some applications of the technique. So basically co-immunoprecipitation, you'll have, for example, protein A and protein B that are a part of the same large complex. And you'll also have an antibody that recognizes protein A. So if you isolate the proteins from the complex and use immunoprecipitation, protein A will be precipitated along with the complex and that protein A is a part of. However, since protein B is also part of the complex, it will be precipitated with protein A too. So that's what makes co-immunoprecipitation. And to detect protein B, you'll use staining of the gel, and immunoblotting can be done. So both of these techniques kind of go together with each other. So with immunoblotting, this is done using SDS page, which stands for sodium, I apologize if I can't say it, dodecyl sulfate. Then they're blotted on a nitrocellulose or nylon membrane, and the proteins have been moved from the gel onto the membrane by an electric current. Once the transfer is done, the blot is washed, incubated with a primary antibody, washed again, and then exposed to a secondary antibody. And finally, the secondary antibody is conjugated to an enzyme. So the presence can be detected using a colored precipitation reaction or chemiluminescence. Sorry if I mispronounced that. And that which means it's able to generate light through a chemical reaction. All right, so the next slide, I was able to find a pretty interesting article relating to co-immunoprecipitation and immunoblotting. So with this research that I found, a key question that was asked by the author, basically he hypothesized that the integrin tail of a glycoprotein 2B-3A regulates the signaling cascade in platelet by modulating the interaction of cytoplasmic and cytoskeletal proteins. So with the experimental design, the aim of this research was to characterize some proteins and investigate their capacity to control integrin functions in human platelets. The proteins that were chosen for the study were PKAR1 alpha, Pinch1, and HIC5. So there's a figure up above in the slide that basically explains the results of the experiment. And the experiment, of course, used co-immunoprecipitation and immunoblotting to confirm the interaction of the three proteins with the platelet integrin. And it showed that neither PKAR1 alpha nor Pinch1 proteins bind to the integrin chain. And they also made a discovery that HIC5 protein binds to the integrin. And based on the photo, you were able to observe that HIC5 binds to SZ 0.22 from resting platelet lysates. Okay, so with interpreting the data a little bit more, Western blotting was used to verify the presence of these proteins in human platelets and examine their capacity to be altered by platelet activation. And we demonstrated that platelets express all three proteins. So by using co-immunoprecipitation, again found that proteins PKR1, alpha, and pinch1 didn't bind to the integrin directly, but again able to observe HIC5 did, and it may be a novel integrin regulating protein. So some strengths with this research were that there were a lot of errors in previous attempts with this experiment. So they decided to make some improvements by using a series of dilutions of the antibodies, do two different experimental conditions, reduced and non-reduced samples, using higher gel percentage, and adjusting the antibody dilutions and loading higher protein concentrations. A major weakness was that due to a time constraint, they weren't able to perform more co-immunoprecipitation experiments with the HIC5 and analysis of the SZ 0.22, but hopefully they can do that with future projects. All right. So a question that I have that's related to the topic is that can the same antibody for an experiment used in co-immunoprecipitation be used in immunoblotting? So in other words, are two different antibodies required? All right, and then here I have the references page. So this concludes my presentation.